little bit rehash what Kavita told us today from a different point of view, in a different way. So I hope, even if there is some overlap, I hope it will be useful. Also, I just realized I probably was too ambitious in writing down my notes. So please stop me when I go too fast or I put too many things in one slide. And anyway, I will jump over a few things because I just wanted to stress a number of points which are useful, and probably there's much of noise anyway. So I'm going to talk about mutations and drift, and mutations, selection, and drift. So this is the same subject as Kavita's lecture, but I hope that, so yeah, from a different point of view. Mutations are, in a sense, a sort of a sea snake, because it is very hard to characterize in a general way. And usually, what I will do too is to focus on some particular cases, and just to discuss a sort of a problems that come when you try to describe mutations and to put them in a context, in an evolutionary context. So this will be a little bit more empirical than what we had so far. We have seen Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium that was discussed by Kavita is very useful because it is a sort of null model in which you know that when there are deviations from Hardy-Weinberg equilibrium, it means that something is happening. So I remind you what it is. Let's consider a population in which you have sexual reproduction and a diploid genome, and assume that you have two variants, say big A and small a, variants allele at one locus, which eventually you have to consider that they are DNA subsequences in the long genome of these individuals. So you might have homozygous genotypes, so big A, or small a, and then heterozygous genotypes, a big A and small a, which is the same as small a, big A. We cannot really distinguish these two strains. So the two chromosomes corresponding to, so we cannot really make a distinction between homozygous and heterozygous. So we have a population size n and we can describe the genotypic composition of this population by giving the frequency of the different genotypes in the population. So we have X of big A, X of big A, small a, X of small a, and then the P, let's call P the frequency of the big A allele, which is given by twice the frequency of the homozygous big A allele plus the frequency of the heterozygous. And then Q is a complementary probability, is that of the small a allele. Then the Hardy-Weinberg equilibrium theorem comes if you make the following assumption. First of all, you neglect fluctuations, and as we have learned from Kavita, that means that you have a very large population. So because the sampling introduces some fluctuations, and these fluctuations are negligible if the population is very large. Second, we assume that there is no selection for either allele. So it's a neutral, all genotypes are, from a point of view of selectivity, selection are neutral. So a fitness is equal to for everybody. And the third, which is very important, is mating is random. Yes, please.
What do you mean by large in different species? For example, bacteria, we can have millions of them, but
yeah, this is irrelevant for bacteria because they don't really mate in pairs. They have a different form of mating that I'm not going to discuss. But okay, large, by and large here you have to consider that the error you make is made by the standard error. So it goes down like at the square root of the size of the population in frequency, because this is really pretty much like a binomial distribution. So this is what you have as a rule of thumb, depending on the precision you want to use. I was making the point of mating is random. So what is called, this population is called the palmictic population. Biologists are really very fond of these words. So imagine, so it is likely, I don't know, in human populations that somebody with blue eyes will prefer to mate with somebody with blue eyes too. So eventually you have, and in this case, how the equilibrium it will be broken. So if there is what is called assortative mating. So here, one of the weak assumptions, if you like, one of the benchstones, and no, the things that you can check via the Hardy-Weinberg equilibrium is whether mating is really random or assortative. But if you make these assumptions, then you get this, that in the next generation, just in one generation, you reach a fixed point in which the frequencies of the genotypes are given by this one. And in fact, the important thing is that I want to get out of this result is basically that if Hardy-Weinberg equilibrium holds, you can forget about the genotype frequencies and just focus on allele frequencies. And this is where the concept of gene pool arose, say the fact that if this is true, then you can only look at what is the frequency of the different allele for each locus you're interested in, and then each individual, say, will pick up these alleles from the pool as for each locus that is in its genotype. From genotypes to alleles frequencies. So just focus on allele frequencies.
How is that encoded in the Hardy-Weinberg theorem?
That you see, the genotype frequencies are determined by P and Q, which are allele frequencies. So once you know the allele frequencies, and if Hardy-Weinberg holds, then genotype frequencies are known. And so the whole, the whole dance is led by the frequencies of alleles.
Right. Okay.
This is what I'm doing. So now we can look at what happens at alleles and what happens at the level of mutations. What does it mean, mutation? And here we go microscopically, really. And in fact, in going microscopically in this way, we are really throwing out of our attention a great deal of very interesting things that happen at the level of mutation. That is, we are just considering, how to say, the small noise in which the mutation can be described by simple single locus substitution of a nucleotide. So, but let us remind that there is much more going on in the genome than simple nucleotide substitution, and I will not have the time nor the competence to discuss that. You can have a major rearrangement, you can have inversions, you can have plenty of things. Now let's look at this thing because it is the simplest thing to describe and gives us an idea what is the mathematics I can tell you. So we know that the DNA is made, on a chromosome, you have a DNA molecule, and the DNA molecule is a very, very long sequence of written in an alphabet of 4 letters, the 4 nucleotides, A, C, T, G. Then, usually, these sequences transmitted more or less identically from parent to offspring. But from time to time, for some chemical reasons, there is a change in the nucleotide in the sequence at one point. So this is called the single nucleotide mutation. And in fact, actually, if you leave DNA alone without, without any maintenance, there will be some sort of spontaneous rate of mutation, which has some direction that I'm not going to discuss. And then you can, according to the nature of the change, these point mutations can be described as transitions on transversions. You know that the nucleotides are of 2 kinds. So you have, I never remember which is which, say purine and pyrimidine. I think that these are purines and these are pyrimidines. And if the interchange leaves the kind of nucleotide the same, is called a transition. And then if on the other hand it changes, for instance, a T in A or a T in G, it's called a transversion. And the transversions have a more important effect, but they are a little bit rarer than the transitions. Now, this is according to the nature. So this is really to introduce you to the kind of language that you are likely to meet in the next week. This is instead a conceptual point. So if you are looking at the coding sequence in DNA, so the sequence which is actually going to be translated into protein, then a point mutation can have one effect or another. If, according to the effect, you can say that the change is synonymous or silent if the amino acid that is produced by the codon to which this nucleotide belongs is the same as in the original case. So you have a change in nucleotide which does not change the corresponding amino acid. Otherwise, it is called non-synonymous or missense. So in this case, you might have a change in the actual protein, and this change can be neutral, more or less, if it doesn't hit on a functionally important point of the protein, or it can be dramatic, can have dramatic effect. But more importantly, you might have a change which is not a point substitution, but the insertion or deletion of one nucleotide or a short sequence of nucleotides. In this case, you have a very important effect in the case in which, for instance, you introduce one, or you introduce or delete. It's very hard to say because usually you don't have diachronic. So the sequence according to time, you usually just have your sampling sequences and comparing them. You don't know which is the ancestral one, which is the mutated one. So this is why insertion or deletion are put together and called indels. Now, if you have an indel with a number of bases, which is not a multiple of 3, then what happens is that whatever is downstream in the translation, so in the reading of the nucleotide sequence when it is translated into a protein, will be read in a different reading frame. We know that the amino acids are read codon by codon, that is by sequences of 3 consecutive nucleotides. So if you shift your frame, then you have in general that the sequence will produce a completely different sequence of amino acids. And this has usually a very strong effect. In particular, what happens usually is that it's pretty probable that a few codons downstreams, you find a termination codon so that a very long protein is suddenly cut at a random point and is no more functional. So indels are usually very prone to be very heavily disadvantageous. So this is reading frame shift. Now, mutation is a stochastic process. It's driven by essentially, so not only physical processes, but also biological processes. For instance, just the very fact of copying RNA, so copying the DNA makes that the initiation of the copying process, there is, say, a sort of machinery which is going on and which is likely to produce mutations with a higher rate than in the rest, for instance. So there are biological aspects and there are also physical aspects that we all know, for instance, radiation or chemicals which act on the DNA and so on. So because they're due both to the effects of the environment on the organism internal workings. And they can be estimated usually by comparing orthologous sequences. Orthologous means that they are sequences which come from a common ancestor some time ago, which sometimes can mean some billion years, or it can be just a few generations ago. Okay. So usually what one does is that one assumes that the mutations obey some simple model and then tries to use a comparison of orthologous sequences to estimate some of the parameters which appear in this model. And of course, so the idea is that you can observe, for instance, a sequence like this one in one locus of a genome of some life form, and then in a population, in the same population, you find an individual, or you find a few individuals which have these other sequences, and you find that these sequences are very similar, so you know that more or less they should be orthologous. You hope so. And then, but however, they have some differences that here you see that there are some stars that here we have a G, here we have a C, here we have an A, here we have a G. And then there is say, a rule of thumb, which is called Jukes rule, that says that the time separation between the two sequences is 2t, where t is the probability, say that, t is the, the time needed for one of these point mutation to arise. And this is, if you think about it, it assumes a very, it has two strong assumptions. So it has essentially one strong assumption, says that the probability backward evolution is the same as forward evolution, is in the sense that you can go back from here and go forward from here, and you simply add twice this t. And so in some sense, this is already assumes that there is some symmetry between going backwards and going forward. We're going to discuss that a little bit. So the idea, and the point is that if you have two close species, what you are doing, you are comparing substitution rates. If you are looking at the same population, you're probably comparing mutation rates. Substitution rates means, what is the difference between substitution and mutation? Mutation is simply the fact that a new sequence arises in a population. Then this sequence can disappear by drift or be maintained in the population. And as long as it is maintained in population, but is not, say, does not prevail totally in the population, you say that this allele, this sequence is segregating. When this sequence, the frequency of this sequence reaches 1, that means that all the individuals in the population that follow in the next generation will be descendant of somebody who had this sequence. You say that the sequence has fixated. So that and in this case, you say that the allele has substituted. So a substitution is the fact that the mutation is going to fixation. If you have two species in which the sequence is close, let's say a given sequence is close but different, then you say that, okay, they differ by substitution. And then you can say, use this Jukes rule to estimate when the two populations were separated. Usually what you do is not going in one allele, say, haha, one locus and say, haha, this is a measure. You try to get that from several loci and then try to guess more or less what's going on. Usually what you have is that if in some loci you have to, say the separation was more recent, and some other loci it was more ancient, and then you try to, say, devise some sort of the evolutionary scenario for this to happen. Now, yes, please.
Got fixated at some point. That doesn't mean that all the offspring of that generation will get the same sequence. I mean, there could still be mutations.
No, they will still have mutations. Yes, sure. Yeah. Yes, please.
The first point, the comparison evaluates substitution rates rather than mutation rates. I guess substitution is a form of mutation.
Substitution is one of the possible fate of a mutation. You can lose the mutation in a population. The mutation can be, say, once there is a mutation, you can have it, keep it as a segregating allele. So it remains in the population, but a frequency which is neither 0 nor 1. Or it can go to fixation. Right? So, and now what I'm going to say, now this is a point, that if it is a neutral mutation, for instance, silent mutations are usually considered neutral, or almost neutral, which is, and then in this case you can prove, and we'll see how to prove it. It's very interesting and deep in a sense, that the rate of substitution is equal to the rate of mutation for neutral mutations. Now, so the estimate of the Jukes, you can use the Jukes rule to estimate the point of divergence of populations or a species, but this estimate is based on 4 general assumptions that are all false. So that the rates are uniform in time, are uniform along the genome, that they are constant in time, they are the same for the 2 branches, and that the equilibrium frequencies of the nucleotides it's important. In a given life form, usually you have a distribution of frequency for the nucleotides that depends on many things. In particular, for instance, depends on the environment temperature. If you have a higher temperature, then you have a GC content has a tendency to be large, to be higher, and the reason is that a GC binding in a DNA is more stable than AT. So in this way, the DNA itself is more stable at higher temperature. So you ask that the equilibrium frequency are the same for the ancestral sequence and the 2 evolved ones that you are comparing. That may be, say, the ancestral life form was living in a different environment, had a different nucleotide ratio. We don't know. You can make models of nucleotide substitution, and I will skip too much of that because this is using the master equation that Vijay is going to discuss tomorrow, I guess. Oh, today? Yeah, okay. So, so this is, you can describe what is the evolution of the frequency, and I just wanted to describe this model, which is the simplest model. So where is it? So the Jukes-Cantor model says, well, all substitution of any nucleotide into any other nucleotide has the same probability. So these probabilities are usually written by alpha. So at equilibrium, you should have that the probability is quite natural. All the frequencies of the different nucleotides should be equal. So it will be 1/4 for all nucleotides. And then you ask, what is the probability that in a given stretch of time, a duration t, you have that a nucleotide J goes into nucleotide I? And you can say, plug this in the equation, what you get is something like that. So you start, of course, from 1, and then it goes down to this 1/4 in a time which is of the order of 1/4 alpha. 4 because of the different nucleotides. So what is the probability of observing the same nucleotide in 2 sequences? This is the probability that there are two contributions to this one. So one contribution is that the nucleotide simply did not change. And the other is the probability that the nucleotide changed in something else and then came back an even number of times. So if you plug this in, you find that this quantity has this behavior. So it's 1/4. This 1/4 times 1 is just the fact that by chance you get that equilibrium by chance to C1. And then you have this time-dependent probability that, of course, is very large when t is small and then goes to 0, which is due to the fact, say, the composite effect I told you of the mutations. So the interesting point is that this quantity is measurable. So if this quantity is measurable, you can define that you can evaluate this alpha t. And then you can, if you have some ways of estimating alpha, then you can go back and estimate t, which is the divergence time of the two populations that you're looking at. So this is the way in which, by and large, people estimate that, I don't know, the divergence from Homo sapiens from the chimp is X millions of years and so on. They, of course, use a more sophisticated approach, but it's basically this kind of reasoning, right? Yeah. Yeah, more or less, yeah. I will do that in a more systematic way tomorrow. So this I will jump off. Reversibility, this is fun for physicists, but it's probably not very relevant. But I just want to go back to the two models that Kavita mentioned, because they're useful then to take into account. So the point is that we often want to describe mutations starting from a given wild type, which is a good starting point. Usually, say, the variability in natural population is large, but it's not so large that you cannot define something like a prevalent sequence, which is considered a wild type, the most common type for the subsequence you're interested in. Then you can try to model in a simple way what is the effect of mutations on the population. One of the simplest cases is just saying that any mutation will produce a completely new sequence, and you are not going to distinguish between sequences, only to say either they are equal or they are different. So you're not putting any structure in the mutation. This is the infinite allele model. I will use it quite a lot because it is, of course, the simplest model. So you either don't mutate, or if you mutate into something completely different, and then you forget about it. The other possibility, so you have no structure, all mutants are different from the wild type, as different from each other. On the other hand, the infinite locus model, or infinite site model, is the idea that you have a very, very long sequence, and each mutation hits a different site in this sequence. In this case, the mutants are arranged in some sort of hierarchical structure. You have one mutation away from the wild type, two mutations away from the wild type, and so on and so forth. Not only, but you can compare them. So, for instance, you can have that if you have two mutants that are different in different sites, you know that they have as a common ancestor only one who was a wild type. Whereas if they have, say, two loci have mutated and then they differed in a third one, then you know that their last common ancestor had the two mutations in the first two loci, and then they were hit by different mutations later on. If you see what, so, and this is the case that we were considering there. Right? So this is why we had this kind of wobble in Kavita's lecture, because the definition of H star that she made at the beginning was a definition in terms of the infinite allele model. And here you have an infinite side model, and conceptually what she was doing is considering that an infinite side model, which is a very good way of looking at it, is like having a sample of the infinite allele model for each locus. All right? And if you assume that this is an independent sample, you can average over the distances and get the same Hg. You see? So in the case of infinite side models, what you can do is that the mutants can be binned in K classes, and classes with K mutations with respect to the wild type. So you start from the wild type and then you can bin them in K classes. Now, well, let's see what happens if you have mutations and selections. So again, we started from population with two types. The selection coefficient S is the difference between the Fisher fitness of the two types, and you have a mutation rate, A goes to B, B goes to A to iterate mu, and then we have an evolution equation of this kind, in which you have, this is X is the frequency of type A, and so the equation looks like that. Of course, if you have no selection, you have mu times 1 minus 2x with the equilibrium at 1/2. If you have no mutation, you have this S time x 1 minus x. You know that there are two fixed points, x equals 0 or x equals 1, and the most stable in some sense, they're both stable or unstable in the same way, but the most stable will be the one in which x is equal to 1. In this case, if you have both effects together, then you find that there is, you can draw the right-hand side of this equation, you find something like that, meaning that there is a stable fixed point at some frequency x star, you can evaluate, and evaluate as a function of mutation rate. It gives, it's a simple quadratic equation like that, and this is kind of curves you get. Now, you can ask, does it optimize fitness? Well, surely not. We know that since fitness is larger for the, would be largest when x is equal to 1, then of course this does not optimize fitness because of the mutation. So mutation will reduce the fitness somehow. Does it optimize something else? Well, it's quite funny that you can define this function. This function has a term which is defined on the population distribution. So distribution of the alleles in the population. And you have two terms. One is the average value of the fitness of the Fisher fitness. And the second one is defined in this strange way. It's a mutation rate times the log of x times 1 minus x. And I call it entropy, and people who have done statistical mechanics know why. But it's just another term. And then you can prove this thing. You just evaluate the derivative of this function with respect to time, and you find that there is, well, there's this term that is very easy, that is due to the change in the average fitness due to the change of the frequency. And then you have the derivative of this guy here that has a strange form. But you see that both of them are proportional to dx dt, and if you look like, you find that you can pull out an x1 minus x here times this quantity squared. This is a positive quantity, this is a positive quantity, this is positive. So this quantity increases to a maximum, and the maximum is reached exactly at the equilibrium point. So we have found what is called in dynamical system, a Lyapunov function for the mutation selection process, which is, and we're going to use it somewhere. Yeah, please. Well, this, I call it entropy within inverted commas. I just made it up, but it's no. Yes, please. Pardon? Oh, it's minus energy. It's just the average value over the population of the fitness, which is equal to, well, it is equal to x. It's on, yeah, it's on the top. Yeah, it's here. So this quantity is not maximal at x star, but this quantity phi is maximal at x star. And of course, it depends on the mutation rate. It doesn't look like the usual entropy we are accustomed to. Yeah.
Yeah, I asked that I know that entropy usually looks like KL divergence.
Yeah.
Is there a story that would show that?
Yeah, this doesn't look like the ordinary entropy. Yeah. But this is actually my point, because sometimes you find in the literature that this is, well, in some sense it's analogous to optimization of free energy, but the entropy does not look like the usual entropy. That's exactly my point. You can generalize that to multiple alleles, and if you do that, the only thing is that you have to introduce this, well, probably, you have to introduce this matrix. Well, I'm not going to dwell on that, and you can define, again, another question. If you like, we can make a special seminar on that. Yes, please.
Please go to the last slide, or just one time. That phi increases in time that we are showing.
Yeah.
So from the division of phi in average fitness and the entropy, can I say that in time, either variety or average fitness, one of these have to be increased? Can I tell it separately from this equation?
I don't know. We separately, in general, if you start from x larger than x star,
yeah,
then you have a high fitness, and then
yeah,
the mutation will pull you down towards x star. If
yeah,
you start with x, which is very small, then,
yeah,
then your fitness will increase until you reach x star. So it depends on the initial conditions.
But one of these two quantity must increase in time, either variety or average fitness.
I mean, one of them must.
Exactly.
Possibly both. I don't know. In the general case, can be both.
Okay.
Log of x times 1, so it's some invariant distribution, and you look at the log of that. Suppose I had multiple fixed points, more than 2.
Well,
X,
no.
X1
Well,
and
say
X2.
in this, say in the case of multiple alleles, in general case, you find that you can define this quantity, a generalization of this quantity, which is here, in a particular mutation model. And then it works essentially in the same way.
So that doesn't have any connection to the fixed points that you get from the...
It's maximal on the fixed point, and you can find that the fixed point is unique. You can prove it. In this special mutation model, it's a special mutation model. It's not general. And, okay. But don't attach too much importance to this. It's only that sometimes you see that evolution improves something. So I said, well, what does improve if you have mutation? I find that this is the function. But this function, as you correctly mentioned, is, I mean, it's something I made up. I don't think that it has any really relevant. And in fact, if you have multiple alleles, you need to have a special mutation model to work it out. Now let's go to something which is more amusing, because it also has some deep implications. This is called the quasispecies model for some historical reasons, but it's in fact a deterministic model of mutation selection balance in a very large sequence space. So you imagine, so I will define it in a scenario in which we have non-overlapping generations, a very large number of alleles. So it's essentially infinite alleles. And you assume that the mutation rate depends in some sense on the distance of alleles. So and then you write an evolution. So for instance, you can imagine that we have an infinite site model. We have the fittest individual is the wild type, so at mutation 0. And then when you go away, one mutation away, 2 mutations away, the fitness degrades somehow, in some way that I will specify. So you can write down an evolution equation. So you look at the composition of the population in terms of, for instance, the number of mutations. So what we are doing here is something like K would be the mutation class. So the number of mutation from the wild type. WK is the fitness of that, is the Wrightian fitness. We are in discrete time. So it's a positive number. For class K. And then we have this matrix, Kj, which tells us what is the probability that in one generation you go from an individual having K mutations to J mutations, which is some structure I'm not going to discuss. And the average value of the right fitness is defined in the usual way like that. And here we have a generalization of the replicator equation, if you like. So we have that the fraction of individuals of type J, a generation T plus 1, is given by the sum over all classes of the fraction of individuals of class K times their fitness times the probability that from K you reach J in one generation. And because of normalization, we have to divide by the average right-hand fitness. Is this equation clear to you? So we have, they first reproduce and then they mutate. And so we ask, what is the probability that at the end of the day we get a guy of type J? Is given by the probability that he has some parent of type K, which has reproduced, and then it has mutated into K. And probably it has not mutated. So there is also a diagonal term. Is that right? Is that clear? Yes, please.
Would you please tell us what is class? What do you mean by class?
Class, so we have classified the mutants, say the wild type is class 0. If there is one mutation in one locus, it's class 1. If there are 2 mutations, it's class 2. If there are 3 mutations, class 3, and so on. All right? Yes, please. What? Well, the only thing is that in this model like that, the fitness depends only on the number of mutations from the wild type. I will give you an interpretation in a moment. So this model arose in the context of speculations on the origin of life. And the idea is that in order to have life, you have to start with some molecules which are somehow able to reproduce themselves. In order to reproduce themselves, they need to have some structure. So they need to have some sequence which satisfies some conditions. But of course, because they are primitive, they have a very high tendency to mutate, and if they mutate, their structure will be degraded, and they will eventually not be able to replicate. So the problem is, how can the self-sustaining, self-reproduction mechanism arise? And now I will point out that there is a problem. Just let me go ahead. I just wanted to know if you have understood this equation like that. Yes, please.
The quasispecies model, the WK, the fitness function, is it frequency independent? And if
frequency independent, yes, absolutely.
Yeah, any specific reason for that, or just a model?
This is the model. This is the model, and it's frequency independent. And then just bear with me now what I'm going to do. Okay, so now we ask ourselves, what is going to happen to this model? And the mathematics is very, very nice and useful. The first thing is you notice is that this would be a linear equation were it not for this denominator, which depends on x itself. But this denominator is there only to allow for normalization of the frequency. So let's forget about it. Let's forget that we go to the unnormalized population vector. This is a vector with non-negative entries. And at the beginning, say, it is equal to the initial composition vector of our population. And then it satisfies a nice linear iteration equation with a matrix, let's call it a T, so it is, we can write it in this way, there is a matrix TJK, and this TJK is a matrix with non-negative entries because it transforms a non-negative vector into a non-negative vector. All right? Only that this vector is no more normalized. But we can normalize it at the end. We don't have to normalize it at every step. So now we have this iteration equation. We can, you see, there is a matrix T. So this matrix has some right eigenvectors. We can decompose, so we can decompose the initial condition into the right eigenvectors of this matrix, such that if you apply this matrix to this right eigenvector, we have the same, we obtain the same eigenvector multiplied by some constant lambda, which depends on the eigenvector. And then we have a very strong theorem, which is the Perron-Frobenius theorem, which tells us if you have a matrix with non-negative entries and with some technical conditions that I'm not going to describe, but we assume that they are satisfied, the largest eigenvalues, lambda 0, is positive, has a unique right eigenvector. It's important it is a unique right eigenvector whose entries are all positive. Whose entries are all positive, not just non-negative, positive. And therefore, if we iterate this dynamics for a very long time, what will end up is that whatever initial state we start, we end up with something which is proportional to this right eigenvector. All right, times some constant here, which anyway will go away when we normalize. Is clear this result? So what is the meaning of this result? It means that if we take the quasi-species model and we wait long enough, iterate long enough, the composition will reach some steady state in which all the types of the possible types are populated, more or less. And the problem is that more or less populated, who's more and who's less? This is the question. But they will all be somehow populated from whatever initial condition you take. All right? So this is what I just said. Now, we have assumed that there is an optimal genotype W0, and then there is, for instance, a case in which all the other ones have fitness which is smaller than this one. And then there is some mutation probability of going from one to the other in any way possible. So I'm lumping all the other sequences except the wild type, or except the 0 type, into some background, and the probability of mutating into any one is given by this mu, and let's assume that mu is, here we have a mu time, is u, in fact. Let's define it in this way. This r is the length of the genotype, and okay. So this is the probability that a sequence mutates into any other thing, and then we forget about the possibility of going back to the original sequence. And then you try to solve for the steady state. For this very simple case, so I remind you, this simple case is that have the wild type which is fit, and all the other ones are not fit. You can mutate away from the wild type, but never back. And then it's very easy. What happens? So how can you get a wild type sequence? Well, first of all, it has to reproduce, so you have x0 times w0, and then it has to not mutate, which is probability 1 minus u. U is the probability to mutate into any other sequence. You normalize, you have to normalize, and this is just a normalization, average value of the fitness. And so you can simplify by, so define W divided by W0 is 1 minus S. So S is the fitness advantage of the wild type. U, as I remind you, is the probability of mutation. You can solve it, you can write down in this way. And then you ask, what is the fixed point? So you end up with this result. X0 star is equal to 1 minus U over S. And then, okay, if U is less than S, it's all okay. You will have a finite frequency for x0. But what if u is larger than s? It gives you a negative number. Negative number doesn't make sense. So what happens is that the only solution in this limit is x0 equals to 0. What does it mean exactly x0 equals to 0? It means that all sequences are equally populated. If you have L loci, you have 4 to the L, if you like, sequences. You have an enormous number, and then the wild type is just one among them. So it has a practically vanishing frequency. Almost equally.
The wild type is not populated, but you don't know about
I'm not saying this is frequency. So again, I will. Let's look more in this case. What I did here is n in this case, unfortunately, is the length of the sequence, and we assume that there is only binary sequences. Mm-hmm. So the total possible number of sequences is 2 to the power n. And then I can solve exactly this equation for a finite value of n, so that you find, for instance, for n equal 50, you have this nice looking curve. But if you increase the length of the sequence, you find that it flattens out. So in fact, here you find that there is a tiny non-zero frequency for the wild type, but this tiny non-zero frequency is of the order of exponential to the minus n. So it is very, very, very small. Whereas if you have u less than s, what you have is a finite number, which, as you see, does not really does not depend much on the value of n. This is the exact result. But, okay, so if you look now at the distribution of error classes, I defined it before, the error classes. So classes 0 is a wild type, classes 1 is 1 mutation, 2 mutations, so on and so forth. So we start from, this is U and not X. So we start from 1, and then this is the curve I showed you before for n equals 60. And then this is the frequency of error class 1, error class 2, error class 3, as you see. You see, all of them essentially have a vanishing frequency when the mutation rate is beyond S, beyond the selection factor. Yes, please. Okay, this is U, and this is S. S is equal to 0.2. So here U is less than S, and here U is larger than S. So this is called the error threshold, because it means that if your mutation rate is too large, then essentially you have no chance of transmitting the information contained in the sequence to the next generation. Okay, this is distribution. So this is the distribution again for a given. But now there is some confusion, and I'm trying to probably add to this confusion. So the fact is that people consider that, okay, once you are gone to through the error threshold, then say in what we say in French is the carrots are cooked, so carrots are boiled, you cannot do anything more with them. And people have also proposed to use this concept of the error threshold to cure some viral diseases. That is, introducing some mutant agents to push the viruses beyond this threshold so that they will disappear. But there is a difference between the error threshold and the extinction. The error threshold is only about keeping the composition more or less aside, but it works in a context in which a population not only is infinite, but it is infinite, and you never discuss what happens to the population in the large. Whereas extinction is something that is related to the size of the population. You have to compare the two. You see, so let's discuss what happens in a concrete example in which we have 3 genotype classes. So we have class 0 is the wild type with a fitness, say, larger than 1, which has a mutation probability U0 to go to the class 2. Class 2 is an intermediate class with a fitness which is lower, but non-vanishing, means that still. And then you have a class 3, which fitness equals to 0, so it's dead. So this is the scheme, all right? So we have the wild type, then we have an intermediate type which is not so good, and then we have less. Now the point is what happens, so you ask what is the evolution for the composition of this system. You can simply write down the thing, but now the real difference with respect to the model, with respect to the equation that I wrote before, you see here we have a linear equation. Why? Because we are looking at the actual number of individuals in each class and not at the composition vector, which is normalized to 1. You see what is the difference? Now I'm looking at the evolution of the actual numbers of type 0, type 1, and type 2. Every time I have a type 2 at the next generation, they simply disappear. So in fact, all the game is played between, all right? And then you ask, well, and this is the transition matrix, so you can figure out. So this, you see, W0 times 1 minus 0, this is a reproduction of 0, not mutation. This is a mutation term, this is non-mutation in class 1, and this is the source of the, so only class 1 can go into class 2. And you evaluate eigenvalues, eigenvectors. I will not do this calculation. You can do it in, yes?
I think the third last entry should be 1. 3 plus 3. Remains as N2 all that.
Well, no, because they die. No? They die. They die.
So what does the number N2 mean? That's the number of dead.
No, so what you have is that class 1 mutates into 2, and the next generation they die. All right? Okay, so you can evaluate that, and so it's the same mathematics, only that this time you have this lambda has an actual interpretation. Before, the lambda did not have an interpretation because it disappeared by normalization. Here, lambda, which is the eigenvector of the leading eigenvector of the matrix, tells you what is the growth rate of the population. Well, the growth factor of the population, because we are in discrete setting. And so if lambda is larger than 1, the population thrives and it actually expands exponentially. If lambda is smaller than 1, you go to extinction. So what you have to look at is where is lambda less than 0. And here we have, on the one hand, the error threshold, and on the other hand, we have extinction. And let's see what happens. So the error threshold is given by essentially the same formula we had before. You have to compare what happens on the wild type and what happens for the, say, the first class. So if the first class reproduces faster than the wild type, then it will dominate, all right? If it reproduces slower than the wild type, then the wild type will dominate. So the error threshold is when this factor is equal to this one. If this one is larger than this one, then the wild type will go to 0. If this factor is larger, then the wild type will dominate. And the extinction threshold is given by this other equation. There are two equations. And then what you get, okay, so we get the transition. So here I put the two values of your get as a function. But say, let's put it in this way. Here we have the mutation rate. Here we have the relevant factor. So one is so this line here. This line here corresponds to the dominant eigenvector for the quasispecies equation in this context, and the other one is the dominant eigenvector for the population rate. So the error threshold arises when this eigenvector crosses the population rate eigenvector. It means that the rate of increase of the population of the wild type is faster than the actual population rate increase, then you are below the error threshold. If it is lower, then it means that a fraction of individuals of the wild type will go to 0. And then when this other eigenvector goes below 1, you have extinction. So in this case, what you have is that you have first the eigenvector, and then you go along this line, which is extinction. So in fact, what you have in this case is very interesting, that you go to the error threshold, and if you did not have if you did not have anything else, the extinction would appear here at some value of the mutation rate. But in fact, because of the error threshold, now you're moving on this other line. And the extinction is delayed because of the error threshold. On the other hand, you can have a situation in which the extinction happens before the error threshold, like in this one. So that means that, in fact, if you want to get rid of a virus, you don't have to think about the error threshold. You have to think about the extinction itself. Well, the error threshold will never happen because the population disappears. But the point is that you have that if you look at this factor here, it goes below 1 at a certain point. That means that in this case you have that the error, okay, the population just you tune by, well, in this case, I fix W naught, and then I change U0. I set W1 equal to 1, I change W naught, I fix W naught to this value, 1.75, and then I change U0. And then evaluate these eigenvalues. Just the value of W0. Yeah. And this is the phase diagram. This is W naught, this is U0. So here we have the order phase in which the wild type is prevalent. And here we have the error threshold. In this region here, you have that the population is not extinct, but the wild type has a very low frequency. And this is the region, so it's called the disorder phase. And this region here, the population is extinct. And what you see is that this curve here is not in the continuation of this one, and this is what I said, saying that the error threshold sort of delays extinction. How long I have? Oh, 20 minutes, I can only... so we have seen the finite, the Wright-Fisher model, and I'm only going to discuss what happens when you have many alleles. So let's say individuals of type K, K1 equals to R, with each of them with some fitness WK, non-overlapping generations. And then we generalize, this binomial sampling formula that Kavita introduced to the case of R possible types. So here we have that the probability that you have a certain composition, ny prime to nr prime, in a finite population of size big N, is given by this multinomial coefficient in this way, times the product of this probability xi I to the power n prime I. And the xi I is given by, in terms of the frequency of type I in the previous generation, is given by in this way. So psi k for type K is given by frequency xk times fitness wk divided by this normalizing denominator, which is actually the average value of the Wrightian fitness for this population. And then you can look at this and you find that this is so you can, for large values of n, but not infinite, is approximated by a Gaussian with a mean n times psi k, and the variance is given by the same formula that we have seen before, all right? And in fact, you can also say there is this is really what you get. So what you see if you iterate this, this is something like that. And in fact, the easy way of looking at these dynamics is the one that Kavita described. Did I put the diagram? No, I didn't put the diagram. So if you have, say, at a certain time this composition, each color is a different kind, and then you want to know what is the composition and the next generation here. And so the idea is that each of one of the new individuals of the new generation picks up at random the parent in the previous one with a probability which is proportional to its fitness. And then, so it picks up and then it copies it. And at this level, we don't have mutations. So, and then you iterate, and let's look at what happens. So this is probably nice. This is a very easy simulation you can do. I don't remember the parameters here. And here we have a neutral case. And we have, I think we have 5 different kinds. And then you see that there are wild oscillations in the frequencies, but eventually after a certain number of generations, which is, we'll see is the order of the size of the population itself, there is one which prevails and goes to fixation. Look at that, this guy who went to fixation here was very, very rare. You see? So you cannot predict what is the eventual fate in anything. Let's look at, so this is, if you run the same model from the same initial condition in several times, and so what you get is, say, these kind of very widely changing results, and sometimes you end up in fixation, so this is extinction. You're looking at different trajectories for the same time. You see, they start from 0.25. I think in this case we have 1 port. Let's look at this case. You see how different it is. So what happens here? Well, this is a Wright-Fisher model with selection. So you see that there is a more or less exponential increase of this. Well, there is a sort of initial wobble, but there is an exponential increase of the frequency of one type, and then so pretty soon it reaches fixation. But if you look carefully, you find that this blue guy remains quite a long time. So here we have a 10,000 population of 10,000, and then the difference is only 10%, and we start with 10 different types, so each with equal frequency. Now, here I just wanted to emphasize one thing, that the fixation is not deterministic. So here I just change a little bit the data in order to have an illustrative example. We have only 500 individuals, and the advantage in fitness advantage is only 1%. So with 1%, you can estimate that there is a fixation more or less with probability 1/2. And in fact, here in this case, Ronald Fisher would have killed me for that. Exactly in 5 cases you go to fixation, in 5 cases you go to extinction for the preferred variant. Right, okay, and this is very interesting. So I just tell you what happens. In the case of neutral Wright-Fisher model, then the time to fixation is proportional to the size of the population. When you have the selection, then the type of fixation, we have just seen it in Kavita's lecture, is of the order of log of the size of the population divided by the fitness advantage, right? So it's very different time scale. And I hope tomorrow to be able to tell you something more about that. So I think I can jump on this. Yeah.
How we can do experiment is over and nothing, and we are in equilibrium or a steady state, and nothing is going to happen in the future. For example, in these plots that you showed, you see that's as you said, the situation is very random, and we cannot. We saw that when some of the classes were going to be distinct, the situation changed and it become fit.
Yeah. But here we are without mutations, and therefore once it's fixed. There is nothing more than to happen. So this is the case. If you have mutation, then the thing runs on forever. You know, new variants are introduced, and they go their fate. So the process never stops.
Do we have I don't know species? I don't bacteria, that's just one of these. One of these models work for it. I mean that just mutation or just selection dominant or we want to, for example, consider kind of E. coli. We have all of these.
Well, this is more difficult. Say, for instance, in the case of bacteria, the size of bacteria's population is very large, but on the other hand, they have a comparatively, say, a certain amount of mutation rate per individual. So that means that if you start having following a colony for several generations, you are likely to see some sort of mutants. Most of them will be neutral anyway, so you will not be able to see them except if you're going to sequence them. But so this, say, by and large, you would treat any lab population of bacteria over a short time as homogeneous. And in fact, this is what people usually do. And of course, if you look, for instance, of, say, the Mendel's peas and so on, if you look at a comparatively small population, but say still substantial, but comparatively small population over not too long stretch of time, you can again imagine that this kind of drift would occur. So this is a good model in which you can neglect mutations and just look at the effects of drift. And it works pretty well.
Another short question. These data are simulations?
These data are simulations, very trivial simulations. Now, I think that I will not be able to introduce you to intricacies of the diffusion equation, and so, but so I think that this is, but I will show you some curves. So this is the solution of the diffusion equation that tells you what happens in the neutral case. So we imagine that you are considering now a collection of populations. This is important, no? Instead of considering the fate of a single population, we assume that we have a collection of populations characterized by the frequency of one allele, and say there are two alleles, and they are neutral in this case. At the beginning, you have a distribution, say, like that. It could be even a delta-like distribution, say, all of them have x equals 1/2, just. But let's start from a normal distribution, more or less normal distribution of this kind, and then let's see what happens just by sampling. What is the fate of the distribution of the frequency of the alleles? Is right? X is the frequency of the big A allele, and 1 minus X is the frequency. So what you get is that you see that the distribution widens up a little bit, but it goes down. Why? I mean, the integral, at the beginning, the integral was 1. Now the integral is less than 1. Why? Well, because some of them got extinct, and some of them got fixated, and this will be represented by a delta function that I didn't want to represent here. So you have that the area goes down because the extreme get more and more populated. But how does it go down? Well, look at that. It becomes flatter and flatter. And after a while, look at this time scale is in terms of population size. You see, this is a number of generations equal to half a population size. It's almost flat. And this is when it is equal to the population size, it is flat. And then it will keep on going down like that. All right? Up. Now, what happens if you have a skewed initial condition like this one? Skewed initial condition, up, goes down. And again, it keeps skewness, but in fact it gets flatter and flatter. And eventually, when t is equal to n, it is more or less exactly equal, exactly flat. What happens now? So the probability that x is different from 0 and 1 decays exponentially with a time scale which is of the order of n. So this is a probability of coexistence, and then, okay. Now you can evaluate, this I will not evaluate, the fixation probability formula is, I will not go in. This is what happens with selection. And this is the shape of the long-living eigenfunction, decay rate, so you can calculate whatever, and this is interesting. This is the fixation probability as a function of selection advantage and population size. We know that when there is no selection advantages, Kavita has brilliantly derived, the probability of fixation is oh, this is not NS, this should be X here. Sorry, huh? This should be X, that is the initial probability, is equal to the initial frequency. Now, as NS becomes a positive, then you see that the curve goes a little bit above the initial frequency. And then when NS increases, it goes more and more to this curve here, so that it is sufficient to have a fraction that is larger than a certain value, which is of the order, which is, you can evaluate, and then I will tell you later. So the probability becomes almost 1, all right? So this is what happens. And this is the fixation probability for a single mutant in the population. The interesting point I wanted to make here on the fixation probability with initial frequency equals to 0, x equal to x0, is that if you take x0 times big N, this is equal to the number of individuals of this variant at a given time. If N is very large, you can neglect this term in denominator. So that means that this probability of fixation becomes independent on the population size, provided population size is large enough, and depends only on the actual number of variants that you are looking at, right? And of the selection advantage. And this is a very surprising result. Okay, so this is the fixation probability of a single mutant as a function of S with different values of a population. See, where the population is very small, you see that there is a non-negligible probability that even a deleterious mutant can fixate. But if N becomes large, then you start not seeing it anymore. But probably on a logarithmic scale, you would see something. So this is a frequency needed to obtain fixation, and this reminds you of something that we have seen yesterday. Now, how large must be X to be almost sure that the beneficial mutation fixes? Well, you have to solve again. The probability of fixation at the X star must be larger than, say, 1 minus gamma. Let's take NS much larger than 1, so we are in the, say, favorable condition. And then you have a P fix is given by this simplified formula. You just invert that, and you find that N must be larger than N star, which is given by absolute value of log of gamma divided by 2S. So apart from a factor of 2 is pretty much the same thing that we had yesterday for the probability of not being extinguished. So it is, so the fate of the mutant is determined in the initial phase in where it undergoes a branching process. We can treat it as a Galton-Watson process, and in which the size of the total size of a population is irrelevant. When it reaches this value, say, number of individuals of this size, then we know that it's pretty sure that it goes to fixation. So this introduces us an important concept. When does a fit mutant get established? A fit mutant gets established when its size is of the order, say, larger than or the order of this quantity, because in that case we can say that the probability that it eventually goes to fixation is finite and is practically 1. But before that, it is a stochastic process, and what is also important is that you can use Galton-Watson process without caring about the population size to describe what happens there. All right? So this is quite surprising, quite nice. Now, this is important. So let's consider now we have a new mutant. It's for a neutral mutant S equals 0. And we have just seen that the probability of fixation is equal to the initial frequency. But the initial frequency for a new mutant is equal to 1 over n. Therefore, if U is the mutation probability per genome per generation, this is also the probability. N times U is the probability that a new mutant arises in the population. But the probability that it goes to fixation is 1 over n. So what we have is that what is the rate in which new mutants go to fixation in the population is given by UN, which is, by UN, which is a probability that new mutants arise per generation, times 1 over n, which is the probability that a new mutant goes to fixation. And so this is equal to U. So this is the result that I had mentioned before. The substitution rate for the neutral mutants is equal to the mutation rate. And in fact, you can generalize it more. And this is interesting. We have seen that the probability of fixation depends on the product n times S, where S is the selective advantage, n is population size. If n times S is smaller than 1, then the probability of fixation is as if it were neutral. So it is equal to 1 over n. So this is also true for slightly advantageous or slightly deleterious mutations, and that will work in the same way. So we have a sort of area of mutations which are practically neutral and in which this substitution rate equals mutation rate holds. And this is also important because this is the way in which you can apply what I mentioned before about the mutations. The mutations are used to estimate the time of divergence since the common ancestor. And you would say, well, but a lot of things can happen. There is a fluctuations in population size and so on and so forth. But if this is true, then that kind of reasoning works not only for strictly new mutations, but also for mutations which are slightly advantageous, slightly deleterious. And I think that I can stop here because otherwise I'm going too far away. Yes, please.
Thank you. Just wanted to read off the numbers. So in the last slide, so for instance,
Pretty close, yeah.
For instance, if you have the human genome versus the bacterial genome, like this for neutral mutant, like the scale, like if use the mutation probability per genome and generation, like, you know, what numbers are we looking at?
A useful rule of thumb is that typically we have a few mutations per genome per generations, and this is more or less independent of the life form you're looking at. This is just a rule of thumb. It's not completely correct. For instance, in bacteria, this is a large exaggeration. Most of the bacteria do not have mutation at all, so probably you are off by a factor of 100 or so. But for higher life forms, is a good rule of thumb. So we have of the order of 10 to the 9 nucleotides, and we have a mutation rate which is per locus per generation 10 to the minus 9 of the order of. Yes.
Can you please go to the first slide, first and second?
First slide.
Very first slide of the Weinberg and Hardy. That
it takes a long time. I don't know how to get all the way down, all the way back. Here.
Yeah, the last class when it was explained the Hardy-Weinberg equilibrium,
Yeah.
then there was a constraint that the total genotype frequency summation will be 1.
Yeah.
So just back the side on one side back.
One slide back.
Yeah. So P plus Q should come as 1. Am I right?
Probably yes. There is a factor
of 2 probably will not be there.
Yeah, factor of 2 missing. Yeah.
Yeah. And second question is again the same question I asked to the last class. Okay, I am calculating probability of fixation. Okay. Now I am saying that this allele is going to be fixed after 1,000 generations. Okay. Now I also know the dynamics, so it should be a fixed point, no issue. Now the thing is that if it is an unstable fixed point, so it is unlikely to be reached. So I cannot connect these two things. Means
because you are thinking in terms of a dynamical system.
Exactly.
And this is not a dynamical system. This is a stochastic process.
Okay.
And in the dynamical system, what you have is a first-order differential equation, which has a right-hand side, which is a function of the observable, and that's it, all right? So fixed points are given by the equation in which the right-hand side vanishes, and around that you take the derivative, and then by looking at whether this derivative is positive or negative, then you decide whether the point is stable or not. Here we have not only, say, a stochastic process. If you describe this as in a partial differential equation, you have that. So let's put it this way. We have a deterministic part and a stochastic part. It turns out that both the deterministic part and the stochastic part vanish at x equals 0 and x equals 1. So it is a singular on either side. So that the concept of stability of dynamical system does not work in this case, because you can simply not do anything, right? So it goes. So you have to be more sophisticated. That's all. Any more questions?